Welcome. My team will be talking about biological computing today. My name's John. Let's get straight into this. So, what is biological computing? Well, biological computing is the idea of using cells, these could be plant cells, animal cells, your own cells, to do computations for us. But how does a cell do a computation in the first place? Well, a cell will take in a stimuli, or a cause, and then produce a response, or an effect, to that stimuli. And therefore, if we can understand the different types of stimuli and the different types of responses for all of these different types of cells in this world, we could then manipulate cells to create computations and get results that we want or desire. But why would we want to do this to cells? Well, we would do this so that we can harness their processing power. Their processing power is a very strong tool in nature because it uses a form known as parallel computing. We want to use parallel computing. Let me give you an example of why we would want to use it. So, if you took an average smartphone of today and created that completely out of biological computing parts, the phone would then be 10 times, if not more, stronger than today's supercomputers. The potential of parallel computing is extremely immense. Therefore, we would want to look into biological computing further. So let's see the applications of it, shall we?
In the present day, the practical applications of biological computing are mostly limited to the medical field. Since most of the cost of programming a cell is in the initial stages, scaling production is only limited by the nutrients and the time it takes to grow cells. Cells could be programmed for autonomous detection and creation of proteins for treatment. The target of these cells could be any abnormality, cancer, viruses, or diseases. But programmable cells are not limited to medical applications. They can also be used in place of the most common computers today, laptops, cell phones, or microchips. Today's silicon-based supercomputers are easily rivaled by biological computers. Not only is the computing power of cells efficient, but the data storage capacity of DNA is very useful, the application of which we will see in the future. Thank you.
Hello, my name is Tim Hannon, and I'll be presenting on the limitations of biological computing. The limitations to biological computing are all centered around the fact that these computers are made from organic materials. Organic materials are hard to replicate and keep homogeneous throughout the system, therefore leading to inconsistent output from different cells. This inconsistency generates the need to monitor all the cells to make sure that they are all reacting to their programming the same. Because the components of the computer are living things, their life looks different than a modern computer. The cells can wear down or degrade, they can die, they can reproduce incorrectly leading to computing inconsistency, and they need specific living conditions to operate. Modern computers react exactly the way they are designed, and if they don't, the fault can be given to the programmer. But when a biological computer doesn't compute, then the issue can be hard to pinpoint, either the programmer or the cell. In all, biological computers are a step in the right direction and have amazing potential in the world of computing.
Biological computing has many uses in the future. However, the technology is still in its infancy, and there are several reasons we would want to implement this over traditional chips in the future. The first is because once the first cell computer has been created, it becomes incredibly easy to create billions and billions of them, with the only inputs being the cell's nutrition and the time spent creating them. In comparison with traditional chips, which require like lots of precious metals and materials such as silicon, these will be far easier to produce since you can just replicate the process billions of times. This technology can be used in a lot of places, however, there are some that immediately spring to mind. The first being medicine, due to the organic nature of the computers. However, as we are approaching the limits on data storage, it may become feasible to use these billions of cells in order to house data. And finally, while it might take a while before it actually occurs, traditional computation is likely possible on these cells, given that there has actually been someone who recreated a logic gate on one of them.
To recap, a biological computer is a living cell or organism that reacts to changes in its environment. By manipulating these changes and detecting the cell's responses, we can use these cells as a sort of computer. Biological computing has potential to completely revolutionize the medical field, especially in DNA centered sciences. Because biological computing is based on imperfect living organisms, it's extremely unlikely that they will ever replace traditional computers as they don't have the same capabilities. They're much better suited for specific biological and medical applications. There are many ongoing research projects about biological computing, but only time will tell if biological computing will become a mainstream technology.
However, some research areas to consider in computational biology are genetic epidemiology, functional genomics, and cancer genomics and evolution. To find genetic risk factors for complex clinical features, researchers in the domains of statistical genetics and genetic epidemiology create new techniques and enhance established approaches. These initiatives have helped us understand the genesis of illness better. To find genes associated with illness and clinical outcomes, cutting-edge analytical techniques are paired with massive amounts of clinical and genomic data. In order to better understand how genetic variations affect illness within and across populations, computational biology's investigations encompass a wide spectrum of common and unusual diseases, including cancer, cardiovascular disease, and more. These investigations aid in identifying those who are more susceptible to sickness through biological discoveries. These investigations also aid in the development of treatments, improving patient diagnosis and care. The knowledge of genetic pathways causing disease risk, symptoms, or prognosis is improved by functional annotation of genomes, which is crucial for interpreting the vast number of individual genomic variants in connection to numerous illnesses and malignancies. The implications of genetic variations are still poorly understood since the majority of them reside in non-coding areas of the genome. How researchers categorize, measure, and make sense of altering genetic fingerprints inside and across tumors over time is the new frontier in the study of cancer. Through the examination of single cells, the geographical and temporal progression of cancer cells is investigated. Finally, cancer genetics and evolution have the potential to completely alter how we think about cancer and how we treat patients.